So I'm old. I remember watching Sesame Street growing up, and one of my favorite characters was the typewriter. Type things out there, roll across the screen, type and go. So I think that is stuck in my subconscious, as I say that way too much. Alrighty. So the good news is we are now into the penultimate chapter here. I don't want this. No, I don't want subtitles. Stop. Right. Okay. How do I turn those off? Anyway, I'll forget it. They're in there. So we're going to talk about protein metabolism. So we did our DNA metabolism, which was a really, really, really long chapter, of which 1/3 of it was just talking about recombination, which is a pain, and I will not emphasize as much stuff. But then we had our RNA metabolism, which is kind of cool. And now we get into our protein metabolism, where it should be a lot of things that you know, with just some added details in there. So we'll talk about the genetic code, the synthesis of our tRNAs, and our translation process. And then at the very end, so in addition to initiation, elongation, and termination, once we have our protein, we have degradation as well too. Okay. So when we make proteins, it requires a lot of energy. Every time we add a nucleic acid in our growing chain, or we start bringing in our amino acids, we're going to need ATP in there. So our protein synthesis can use something like 90% of the energy in the cell. A lot of this will depend on how many copies of a particular protein are made, and that will, again, ideally correspond to how many are needed. So that if we need this protein for, say, growth or development or differentiation or signaling or structure or whatever, the body is going to try and make the number that it needs. Our proteins are also targeted to specific locations. We will talk about signal sequences there as well too, which are just kind of like, you can think of it as like your bus ticket or your train ticket. This is my destination. This is where I'm going to. So we have these signal sequences that will tell us where the protein belongs. And it's really kind of neat because you can do experiments where you can like swap out signal sequences and send proteins to the wrong place or whatever. And then on the other hand, our degradation is going to keep pace with our synthesis. So again, the whole idea of homeostasis, as a protein gets targeted for degradation, a new one is being made to take its place. Okay. So our protein synthesis is very complex. We've got tons of ribosomal proteins and activation proteins, and basically like 300 different proteins or biomolecules. I tend to say proteins, but we have our tRNAs and our ribosomal RNAs as well too that are in there. One of the reasons that makes it complex is we often have a lot of post-translational modification that occurs. So that final, and as well as our final processing as well too. So we may be cleaving off parts here, or changing an oxidation state for maybe our cysteine residues to make disulfide bridges. But there's a ton of enzymes that do all that. So if we look at how we first started to understand protein synthesis, we figured out initially that proteins are synthesized on the ribosomes, that amino acids are activated by attachment to tRNAs. So we have this amino acyl tRNA synthetase, which will take our tRNA and match it up with its appropriate amino acid. And then the tRNA itself acts as an adapter. It has a region on it that will recognize the messenger RNA and bring the corresponding amino acid into position in there. If we take a look at an electron micrograph of the ER, you can see all those little dots along the ER. Those are all our ribosomes that are actively synthesizing protein in there. So you can see it as an ongoing process. Big old factory work. Here we are in Winston-Salem, and I can, it's like, we go a little further south too, the textile industry, and constantly making all this stuff in there too, or drying up our tobacco or whatever. Anyway, trying to be local. All right. Early history in here, it's only been like, 65 years that we've kind of figured out a lot of this stuff, figuring out that the ribosome is the site of protein synthesis. And I know it sounds like a long time now, but it's like 65, it's not that long ago that this really all started. Biochemistry really kind of became its own thing. And initially you had biochemistry for like beer, and then maybe like you'd see a lot of biochemistry that was looking at, say, like wool fibers and stuff like that. And then you kind of figure out some stuff, some enzymatics in the 20s and 30s, and then you get to the 50s and there's this big leap, and then it just starts building from there. And if you look at the number of journals that are dedicated towards biochemistry, you go from like 1, 2, 5, 10, and now there are hundreds that deal with that. And it's really just become a foundational field for so many other sciences. Anyway, 56, we get to that whole RNA is like DNA, but didn't quite understand why. And you can figure out that the amino acids are attached to the tRNAs. And 60 years ago, figured out that the what messenger RNA is. Okay, so the neat thing that we have here is we have our 20 amino acids. Okay, we have our 4 bases, our A, U, C, and G in RNA. So we can't code for all 20 amino acids with just those 4. If we use pairs of them, well, 4 squared only gives us 16, so we still don't have enough combinations for all the different amino acids. So we use a 3-letter code that gives us 64 possible variations, and that is enough to code for all 20 of our amino acids that get incorporated into proteins. We use a non-overlapping mRNA code with no punctuation. So what that means is we just go in sets of 3, and we get to the end. There's no interruptions or anything like that in there too. So we just every set of 3. Pictorially, here's what we see. We have our tRNA that functions as an adapter. On one end, it binds a particular amino acid. On the other end, it recognizes a particular triplet codon, which will correspond to that amino acid in there. Okay, so non-overlapping, we basically just go in sets of 3. Overlapping, we might have like 3 different reading frames that we have in there, and that's one of the things we can talk about. So we could read that initial sequence as first codon AUA, second codon CGA, third codon GUC. But if we have an overlapping code, we could have AUA, we could have UAC, we could have ACG. Okay, so we use a non-overlapping code. Right. One of our biggest advances was to actually understand the genetic code. And a lot of this was done through some pretty cool experiments. We could radio label amino acids and then feed them to E. coli, and you only get this phenylalanine. So they use this poly U tail there, and UUU codes for phenylalanine. Right. Somebody else did alternating one where we had like AC, 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 AC. So wherever it started, it was either ACA or CAC, and they coded for histidine and threonine respectively. So they could kind of figure that out there. Okay. So eventually we were able to break the whole code and figure it all out, which we'll see here in a minute. Again, our code is written in 5' to 3' direction, just like we write our DNA sequence, our primary strand. But the third base tends to be less important. It's often known as the wobble base because you can see that oftentimes you can change that third base and you'll still, all 4 codons might still code for the same amino acid. Not always, but sometimes. The very first codon establishes the reading frame. So if we throw off the reading frame, if we have like an insertion or deletion, we can have everything out of order. And that's one of the ways that you can end up with some mutations that kind of can be very, very bad. Sometimes they're not so bad. 61 of the 64 possibilities code for amino acids, and 3 of them are termination codons. So they are the stop codons, and those are UAA, UGA, and UAG. AUG is our initiation codon. It's also our methionine codon, or one of them at least. So if we look at it here, we can see all our possible sequences. So if we start with U and our second letter is U, our third letter can be UUU, UUC, UUA, UUG. And you can see whether we have U or C here, we still get phenylalanine. If we switch that to an A or G, we get leucine. So that's really kind of pyrimidine versus purine in there. In some cases, like for example, if you start with a C and your second one's a G, it doesn't matter what the third one is. It's always going to be arginine. Right? And in fact, even AGA and AGG will give you arginine, but AGU and AGC give you serine. This is actually a really kind of neat aspect because we can look at some different types of regulatory control here, depending on which set of codons we're going to use. So we have some built-in redundancies. You will note that I think AUG is the only methionine codon in there. Yep. And then, of course, we have our 3 stop codons. Okay. So if we end up with, methionine tends to be, this particular sequence tends to be very important because there's only one methionine code in there. I'm trying to see if there's any others. Like everything else, I think has at least 2 for cysteine, 2 for tyrosine, so on. So anywhere between 2 and 6, except for methionine. All right, most amino acids have more than one codon. Tryptophan is another one that only has a single codon. Yeah, I knew I was missing another one. So yeah, we can see that there. Here we can look at the number of codons for them, and anywhere, I guess, between 1 and 6 with 3 stop codons. Okay. We talk about the genetic code being universal. Put a little asterisk up there. It's universal, but not quite. It's used by all organisms, prokaryotes, eukaryotes, so the code is pretty much the same across all of them. Mitochondria are the exception because they have their DNA in a slightly different code. Instead of being a stop codon, UGA is a tryptophan codon for vertebrate mitochondrial DNA. And then you can see our AGA, AGG is a stop codon instead of arginine for vertebrate DNA, mitochondrial DNA. That's a nice little typo in there. Mitochondria also have their own tRNAs. They use 22 tRNAs instead of 32, so little differences in there. So mitochondria is the exception, but all organisms, if you look at their nuclear DNA and all the codons that are produced there are the same across species. So it's kind of neat. I'd mentioned that idea of a wobble base. You can kind of get this nice wobble pairing between the tRNAs and the mRNAs with that third position in there. So some tRNAs will have a nosizate, and that can hydrogen bond with UC or A. These are weaker H bonds, and that's why they kind of call them the wobble base pairs, because they're not as strong hydrogen bonding wise. They're a little bit weaker, but that allows for a bit more flexibility. Let's see here. And so in yeast, our tRNA has the anticodon of GCI, and that will pair up with CGA, GCU, CGC. And again, it's important to note that we're writing this kind of backwards so we can show that matchup. We would normally write that as ICG. All right. So how do we recognize these codons? Again, this is our base pairing here. Our codon sequence is complementary to the anticodon. Our anticodon is in our tRNA. Right? So the codon in the mRNA base pairs with the anticodon in the tRNA. And we have, of course, antiparallel because they're going 5' to 3' in opposite directions in there. So just similar to our DNA with its complementary 2 strands, here we just have a short 3-strand version that's complementary, and our tRNA will match up with our messenger RNA. Okay. Yeah, we can go into details about the wobble base, but yeah. All right. The neat thing about this is our genetic code is very resistant to mutations. We can still see mutations that happen, but we still code for the same thing. For example, if we go back to that, what was it, CG, whatever the third one is, it's going to be arginine because there are 6 ones in there. So we have a silent mutation in there. We basically get a different nucleotide in the DNA, but we still get the same amino acid in the protein. So our genetic code has changed, but we're still making the same protein, the exact same thing in there. So mutations in the first base will usually end up with a conservative substitution. So for example, if you have a GUU becomes AUU, that converts a valine to a leucine. What that means by conservative mutation, valine and leucine are very similar, both structurally. They're both hydrophobic. You're not going to see like a mutation in the first codon that's going to change an alanine to an asparagine, where you go from a small, uncharged hydrophobic amino acid to a large, highly charged one. So we get that conservative substitution in there. I want to make political jokes, but got to resist because we're in the home stretch. All right. So some mRNAs get edited before our protein synthesis. So, of course, we talked about our exons and all that good stuff in there as well, too. But we can alter or add or delete nucleotides in our mRNA. And there are various guide RNAs that will do this. They'll temporarily hybridize and act as templates for editing. So gRNA, you can press your friends, like, what's gRNA? But that's one of our little roles in there. Our guide RNA will bind, and you can see here we have a GU pairing, a couple of these GU pairings that are mismatches, but the rest of it is sufficient for it to bind, and that can allow our start of our editing procedure. We also see deamination reactions that will occur. For example, if we deaminate our adenosine here, so we're going to lose our amino group up here in the base, we end up with inosine. Okay, so we replace that with our oxygen. Same way, our cytosine can be converted into uridine as well by deaminating it and replacing it with a carbonyl group. Okay, so RNA editing often will involve this as well. Okay, so that's all our background. Just flying along. So much for like class discussions this semester. It's like, oh, looking forward to having a semester with the right number of weeks and everything. We've got 5 stages of protein synthesis, and if you are particularly sadistic, you can compare these to the 5 stages of grief as well too. Maybe that's a good mnemonic in there. But the first thing we have is activation. So that's kind of like our pre-step. We gotta acylate our tRNAs. So our tRNAs are out there, and it's like I can hold an alanine. So they had to get charged by having an alanine covalently attached to them. Then we undergo initiation, just like we saw with DNA and RNA metabolism, and then elongation and termination. So our mRNA and our amino acylated tRNAs bind to the ribosome. We start seeing those tRNAs bring in their amino acyls. They read the mRNA, and then the amino acids get attached to each other to make a growing protein strand, and they keep going until you hit a stop codon. The termination, that stop codon signals the termination in there. It's not always a stop codon because some other things can happen that will cause termination, and then it'll fall off the ribosome, and we can recycle stuff again as well. And then finally, once we have our protein that's made, it has to be folded. It has to undergo post-translational processing, maybe some modifications and stuff in there. So, these are our 5 steps kind of listed in order, a little more complex than our DNA metabolism and RNA metabolism. So you saw with DNA, it was like initiation, elongation, termination. For mRNA, it was initiation, elongation, termination, degradation. Here we have activation, initiation, elongation, termination, processing. All right, so there we go, our 3, 4, 5 steps. Okay. So if we kind of look at this view here, we can see our ribosome. We're going to bind our RNA, and then our large subunit will come in and bind. We'll find our start codon. We'll bring in our first charged tRNA, and then we bring in a second one for the next codon that's in there. The ribosome will attach those together. We end up with a growing chain as our deacylated tRNA will kind of, as our ribosome processes along here, our deacylated tRNA will kind of slip out the exit hole. This will slide down here, holding the protein strand and chain. A new charged one will come in, and we'll just repeat this process for our elongation, and then eventually we'll hit a stop signal, and our ribosome will dissociate. We'll release our mRNA, we'll release our mature protein, which can then be folded and processed as needed. Whew, good times, yeah. All right, we can look at this in bacteria, and again, compare this in eukaryotes as well. So we typically see, all the things that are going to happen in here. This is what we need. I'm going to sneeze. Maybe, maybe not. I swear I should pause recording. I should edit my recordings, huh? Take out the ums and ers and what you get. Keeping it real. All right. So our ribosome is the key player in protein synthesis. If you look at bacteria, the ribosomes are, even though they're really, really tiny, they're like 25% of the weight of the bacteria, excluding water. Ribosomes tend to be about two-thirds RNA and one-third protein. The ribosomal RNA is the core, and we have our peptide bond formation in there. The RNA does the catalysis of that peptide bond formation in there. For bacteria, we have two subunits, the 30S and the 50S, which indicate their molecular weights. You'll see something similar for eukaryotes, where you have the 40S and the 60S. So it's just a little bit bigger. Excuse me there. So here we look at our typical structures in bacteria and in yeast. The important thing is there's a little cleft in here. So you have your RNA binding site, and then your cleft where your tRNAs can kind of come in, and then your growing protein in the top, exiting out the large subunit. Okay. So if we look at our ribosomal RNAs, we tend to think of any nucleic acid as a long strand because DNA is a long strand and our mRNA is a long strand. Our ribosomal RNAs have some really complex structures. They're single-stranded, but they interact with each other and they fold over on top of each other, and we can get some pretty crazy looking ribosomal RNAs in here. So you can see all these kind of little hairpins and various structures that show up in here. All right, but the shape tends to be highly conserved. So even though you can see we have this little cruciform section down here that's conserved across species, we also have this little particular key-like sequence up here, and we'll have a little arm that comes off as well too. And you can see our different sizes of arms in here. So again, for a universal code, you would almost expect our ribosomes to look very similar across species. Again, here we go, we compare, oh, sorry, it's 80S and 60S instead of 40 and 60. So yeah, 50 and 70 and 60 and yeah, whatever. Anyway, we end up with our, no, it is 40, 60 for total thing. Yeah, okay. Just the way they add together. But again, they're very similar in structure, and they function pretty much identically. All right. Let's talk about our tRNAs. And this is where you should absolutely watch our protein synthesis, that little Stanford mime of the dance thing from 1970 that I posted a YouTube link to on Canvas. It's awesome. Get popcorn and laugh at all the mind-altering drugs that your grandparents did, because it's pretty crazy. But our tRNAs are, again, RNAs are weird. I kind of like RNAs because they're really unusual. So we can look at our tRNAs. They're going to have some similar characteristics, and we see that they're typically between 73 and 93 nucleotides, and they have a clover leaf structure in two-dimensional shapes. So what that means, we can say they actually make kind of a big cross. So if I got my arms out to one side, and I got my head and my feet, and then what's going to happen is they're going to fold over in 3 dimensions to make this kind of twisted L, okay? And that's going to be the active 3-dimensional form. Most of them will have a guanosine at the 5' end. They will all end in that CAA in the 3' end. And tRNAs have tons of modified bases, pseudouridine, various methylated components, and so on and so forth here. Okay, that ACC is our amino acid arm in there. That is going to hold the amino acid. Basically, whatever amino acid we see is going to be acylated onto this arm. We have our anticodon arm on the opposite side here in our 2-dimensional structure. And this will be the one that will recognize the mRNA, so that corresponds with whatever amino acid is acylated in there. We have our D arm, which has so named because it has dihydrouridine, and it helps with the folding of this. We also have our pseudouridine arm here. Our T5C arm contains pseudouridine, and again bonds between the bases and the ribose and helps in folding in there. And then we have this little extra arm in here that's variable in size. This little extra arm can play an important role, but it's not present in all tRNAs. So, we look at this and say, well, how much of this is really absolutely necessary? Not very much. This folding really helps um, make sure that our binding is very, very specific, but we can pretty much cut out all the middle in here as long as we have our amino acid arm and our anticodon section here. We can get a functional tRNA, right? If we look at our three-dimensional structure here, once we take that cruciform and fold it over, we get our twisted L, all right? Okay. All right, so again, here's our 5 stages: activation, initiation, elongation, termination, and folding. So what's the first thing we do? Is activation. We need to charge up our tRNAs. So we basically have our tRNAs in there. They have that ACC end, and we need to attach an amino acid to it. Okay? So our various tRNA synthetases will attach those amino acids, and we make our amino acyl intermediates. Okay. We also end up producing pyrophosphate, and we'll see what happens with it as it goes through. All right. So here we have our amino acylated end. Okay. So now we need to transfer our amino acyl group to our tRNA. Okay, so our tRNA synthetases work to do this. We look at our mechanism in here, and we can bind this on again through that phosphodiester bond in there. Our various amino acyl tRNA synthetases. Each enzyme binds a specific amino acid and it's matching tRNA. So if you have, say, arginine, so you can have argino amino acyl tRNA synthetase, or you can have alanine amino acyl tRNA synthetase. So the crazy thing here is some cells have fewer than 20 synthetases, right? So in that case, one amino acid gets converted to another after you charge it. So if you go back to that protein metabolism, the amino acid metabolism chapter, and now you can see how some of them can be converted to other proteins. That'll happen after it gets charged. So we might put a phenylalanine on our tRNA, but then we'll convert it into, say, tyrosine or something. Okay. All right. So some people have referred to this recognition of our tRNA synthetases for their tRNAs and their amino acids as a second genetic code. So each one has to be matched up correctly. We don't want to put an alanine on an arginine tRNA in there. So we have this very specific recognition in there. So the funny thing is, very few nucleotides in TRA are involved in the binding specificity, and this is our anticodon region. And then we have the single guanine, uridine, and the amino acid arm in there. Okay, so as long as we have just a little bit of this in here, this is our typical nucleotide position. We typically go from 5' to 3', going around this way. But one of the neat things, we can cut out. So in this case, what do we cut out here? Something like 53 nucleotides. So this is a 76 one. This is only 23 nucleotides long, and this will still work. So we still have our ACC amino acyl binding site, still have our anticodon down here, and as long as we have this single GU interaction that helps give it its shape and binds it together there, this will actually function. It'll recognize. So this is, I guess, alanine, and we can amino acylate this little tiny nubbin of a tRNA with alanine. All right. So our initiation does vary between our bacteria and our eukaryotes. So let's talk about the bacteria first. Our first tRNA is going to be unique. Again, we're going to have this AUG methionine in here. There is a little funky difference here. We have one codon for methionine, but there are 2 tRNAs for methionine. Okay, so in bacteria, you'll have this informal methionine. So you'll have this alternate tRNA synthetase here that will actually use this formulated methionine. And if we have something that's inside, if you need a methionine in the growing protein strand, you would use the other tRNA that just has regular methionine. Okay? For eukaryotes, They start with methionine, not this formulated methionine, but it still uses a special tRNA for that initiation. It's like makes me think of like little kids when they're going into preschool, and you have, like what little kid wants to be the line leader. That's the special reward of the day or the week or whatever for somebody who's good, or sometimes they share it in there. But we have a particular line leader for eukaryotes. Okay, and our formal line leader, I guess they wear a tuxedo, ha 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 ha, for bacteria. You got to have a sense of humor about all this stuff, right? Yeah, so I am the first methionine. See my tuxedo because I'm formulated. All right. Anyway, once we have all of our tRNAs charged, we are ready to start building our protein here. So we see, again, our 20 amino acids that we've talked about before, but we also have a couple others in there that are oddballs, our selenocysteine. So we charge the stop codon with a serine, and this works in both bacteria and eukaryotes. This is a very, very low level tRNA. So we don't see a lot of selenocysteine, but there's a little bit of it floating around there too. We also have this pyrrolysine that will recognize a different stop codon, but this is only in archaea, not in bacteria and eukaryotes. Okay. So here you see, this is what they look like, and you can see they are kind of funky looking. Okay. All right. So our initiation requires a big assembly, big production in here, kind of like our graduation. We have big tents and everything on the field and everything. Got to put it all up and tear it all down. So we're going to have these initiation factors, which are conveniently abbreviated IF1, IF2, IF3. Yay, that's good, right? We're going to see this nomenclature carry over into eukaryotes as well, too. We're going to have a lowercase e for EIF1, EIF2 for eukaryotic initiation factor. The initiation factors play particular roles. For example, IF3 helps keep the ribosomal subunits separated initially so that our mRNA can come into that cleft and bind, and then we can clamp it back shut. So, we'll have this initial codon as well, too, gets guided to its correct position through that Shine-Del-Garno sequence in the mRNA. That's complementary to the mRNA. It kind of says, park here. This is the space where we want to be. Once we have that happen, we're going to see that our formal methionine tRNA will bind to its peptide site. So we actually have 3 sites inside the 30S unit. Okay, and you can see IF1 is in there for initiation to keep them apart. IF3 keeps them apart. IF3 kind of blocks that to get it started in there. And then IF2 will encourage the first codon to come in here. So we have 3 sites, the A site, the P site, and the E site. All right, so I guess I always saw these as the addition, the peptide, and the exit sites in here. Okay. All right. So now our second part, we have IF1, IF2, IF3, and our tRNA with our formal methionine in there. Okay, we will dissociate our initiation factors and allow the large subunit to come in and clamp down over top of that. Okay, so now we still have our cleft, but we now have it loaded, and we have our first tRNA, our first charged tRNA in there. Okay. All right. And you can see here our Shine-Del-Garno sequence and our initiation codon. Right. So we have a funky, this one variation of E. coli has a GUG for our initiation site. Variations. Okay. Eukaryotes, very similar, but instead of having 3 initiation factors, there's at least 12. Has slightly different mechanistic details, and we also see the circularization of the mRNA that occurs in there. We're not going to sweat that, but essentially it does the same thing to load the mRNA, bring in that first tRNA, and then allows the larger subunit to clamp down in there, right? Linker proteins, yeah, put all that in there. So you see we've got this nice little funky loop that happens, and then once all that happens, we lose our initiation factors. But the end result is that we have our mRNA in its position with that first codon bound, and our empty exit site and our empty addition site in there. Okay, so for elongation, we're going to bring in our next charged amino acid. So we will see our anticodon site in there. We bring in the corresponding tRNA, charged tRNA, and then that will, when it binds with its anticodon site to the mRNA, that will put its amino acid in position to be catalyzed, to be combined with the methionine that is there. Okay. And then once this occurs, this complex helps bring it in there. Once it's bound in there, our EF, elongation factor TU-GTP, will leave the ribosome. So you can see we're spending some energy here because our GTP is being dephosphorylated to give us GDP. Okay, so once we have that second charged tRNA in there, we can make the peptide bond. So one of those is on the P site, one of those is on the A site. The N-formyl ethionyl group gets transferred from its tRNA in the P site to that amino acid in the A site. So and we have a ribosomal RNA that catalyzes that. So now our P site has an uncharged tRNA in it. In this case, for our very first reaction, methionine. So you can see here's what's happening. Our amino acid number 2 is going to have a nucleophilic attack of our methionine. So we see that bond change, and we make this growing strand here. And when that happens, our tRNA that becomes uncharged, got a little conformational change and flip over the other way. So we're kind of, I wish I were a better animator. I could try and animate this. So here we have our A site, our P site. I got to find my most excellent tool ever. There we go. All right, there we go. So need 2 of them really with Velcro on them. But basically, A site, our P site, we pass this over to make our amino acyl bond, and then this is going to have a conformational change. We process along our RNA. This now moves to the E site. Our growing strand moves to the P site. Our A site is open. Our tRNA will exit out of the E site as a new charged tRNA will come into the A site. Okay, and we'll just see this growing strand that will continually add amino acids to the amino acid in the A site, which will slide on over into the P site, and that growing protein strand will exit out of the top of the ribosome. Okay. All right. Yeah, we got a bunch of elongation factors for eukaryotes, very similar to bacteria. There's just more stuff going on. Okay. Big difference here is eukaryotic ribosomes don't really have an E site; they just get released from the P site. So bacteria have that APE, and eukaryotes just have AP and a whole something in there. Okay, so we get to termination time. We'll find a stop codon. All right, so here we are in stage 4, termination. 5 stages of grief. No. So what this will happen is we will see this activation of termination factors, release factors 1, 2, and 3. They help hydrolyze the terminal bond in there, release the peptide and the tRNA from the ribosome. So basically we have to take our ribosome subunit and open it up again to let the RNA out. So our release factors help do that. All right. Sometimes we can have a damaged mRNA, and we can have a nonstop complex. Makes me think of Kraftwerk. It's like Trans-Europe Express. It's nonstop. Okay, sorry. Wow, we're getting obscure there. But we can have a translation rescue operation to basically kind of rescue this stalled complex in there. And translation will continue until we get the stop codon and our tmRNA, and then we can degrade our defective mRNA and the polypeptide because it's wrong. So we spend all that energy and then we just destroy it because it's wrong. Perfectionists. All right. More features in here. Lots of energy involved in this. We spend so much ATP for each step of this process in here. But This can also be pretty fast because we can have clusters of ribosomes that we're going to call polysome. So we have one mRNA, we will have multiple ribosomes chugging along on that mRNA. So we got, as we chug along there, we're making multiple proteins as we go. So you can see, for example, here, we're just initiation, and then we're elongating down here. We're getting growing chains of protein in here from these different ribosomes that are all going along that same strand of mRNA. All right. Our last stage, post-translational modification of our proteins. We got to fold them. Some, especially small proteins, will spontaneously fold into their mature shape. That's good, but many of them will need chaperone proteins to help them achieve their mature form. We also typically will see our proteins, we may have a signal sequence on it that says, go to the mitochondria or go to the nucleus. So once they get there, we'll typically cleave off that signal sequence, okay? Many proteins are glycosylated, so we will see the attachment of our various carbohydrates, and some things need to be activated. If you think of like our pre-pro proteins, you cleave something, you get your pre-protein, and then rearrange that and you get your mature protein in there. We looked at insulin, I believe, as an example of that. Common post-translational modifications, adding phosphates or adenylate groups or isoprenoids or stuff in there. Farnesylation. It's always fun to say because I always think of Farnsworth and anyway. Ribosomal profiling. So you can figure out what mRNA is currently being transcribed. The neat thing here is you isolate the mRNA and then subject it to RNase. Any exposed mRNA will be degraded by the RNase, but if it's actually bound to the ribosome, you'll have a good 20 bases or so that are protected by the ribosome, and they won't be degraded. So then you can wash away the nucleases, release it from the ribosome, and then sequence your RNA there, and you can figure out, hey, what RNA is being made currently. Okay. A lot of biomolecules can target translation as well too. It's a very effective way. If you look at various toxins, if you don't want a bacterial infection, or if you don't want something eating, if you're a plant like a particular mushroom in here, you can target the protein synthesis of the parasite or bacteria or whatever it is, that predator, and inhibit protein synthesis. And of course, if you inhibit protein synthesis, that's a good way to kill them, or at least cause enough damage to dissuade them from munching on you if you're whatever there. You look at our mold, puromycin, nice little antibiotic that functions this way. What we have here, tetracyclines, very common antibiotic, cyclohexamides, streptomycins, our various mycins. Look at that as well too. If you look at toxins, things like diphtheria toxin, it catalyzes this ribosylation of diphthomide. So it inactivates it, which leads to an accumulation of fluid in the lungs. So that is not good. Ricin, you are probably too young to recall the subway attack in Japan, but there was it the something dawn, some cult, and they were considered a mostly harmless religious cult until they collected a whole bunch of beans and made ricin, and released ricin inside the subways, and ended up killing several people and injuring a lot more. And it was it was a wake-up call for homemade biochemical danger. You can make ricin pretty easily. We should do that in lab. No, right. Always fun stuff. First, we'll make some methamphetamine, then we'll make some ricin. Okay. Yeah, fun stuff in lab. All right. So the last little section of this chapter talks about protein targeting and then protein degradation. So how do we get a protein where it needs to go? It might need to, leave, they're all made on the ER, but then they got to go someplace in the cell. Sometimes you got to send them outside the cell, someplace else like that. So most of them will have some type of signal sequence that's near the N-terminus, and that signal sequence will indicate where they need to go. It's like, you've got a golden ticket, go directly to the brain. I don't know. This is primarily in eukaryotic cells where our subcellular subcellular organization and our different compartments or our different organelles that we have are very prominent, not so much in bacteria, which are just localized groups within one big bag. So for example, if we take a signal recognition particle, we can direct things to the endoplasmic reticulum. And we can see some modification that occurs here. Your glycosylation will occur in the ER most often. We see some more finishing in the Golgi apparatus. Here we have transport vesicles. So we'll see this budding off from the ER that'll take things to the Golgi apparatus for more processing. I like this. Proteins here are sorted in ways that are poorly understood. Yeah. There's a lot more to learn. There's always more to learn. In our secretory pathway, we can look at proteins for the mitochondria and the chloroplast will bind chaperone proteins to get across the membranes. Oops. All right, so here we can see how I direct them and various recognition sites, all that good stuff here. Glycosylation is very important to the function. And you see a glycosylation pattern of proteins also important for recognition and interactions. So there's two main types: N-linked, where we attach to a nitrogen. This nitrogen is going to be in the residue of the either asparagine or arginine. Starts in the rough endoplasmic reticulum. O-linked will bind to either serine, threonine, or tyrosine because those are the ones that have hydroxyl groups on their side chains. And this typically occurs in the Golgi for proteins that don't go to the ER. Proteins are sorted out in the Golgi and sent on to their final destinations. Always makes me think of those cheesy movies. They're going to die because eventually we're going to degrade them. Okay. Here we look at our glycosylation patterns, again, inside our Golgi. How do we import them into the nucleus? Many proteins will have a nuclear localization signal, and this is not cleaved. So why is it not cleaved? Well, you've got to think if your nucleus is undergoing meiosis, it's going to open up at some point. You're going to release all these proteins out, and you get our nuclei. So you want those proteins to be able to be imported back into there. So you can take them. So that nuclear localization signal is a little bit different. It's not cleaved off after the proteins get to the nucleus. There have actually been some studies out there where we figured out what the nuclear localization sequence is, and then we've mutated a gene to put a nuclear localization sequence on a protein that doesn't go to the nucleus, and you can actually see it'll get transported to the nucleus in there. Targeting, I don't want to talk about these. Yeah. Not too much here. Again, signaling sequences get read by other proteins and partners and associated stuff to get them where they need to go. You can see they have cool names like important because we got to import it into the nucleus and all that good stuff in there. So a lot of times we'll see these big complexes that are made. Speaking of big complexes, when we degrade our proteins, we're going to recycle them or do something with them. For example, we can say, proteins that will stick around for seconds to months. Hemoglobin tends to be around for a very long time. Our defective proteins tend to get recycled very, very quickly. All proteins, though, eventually get degraded. Our mechanism here is typically ubiquitination. We attach this ubiquitin molecule, so named because it's present everywhere. So ubiquitinated proteins indicate this is a target for destruction. This is where I'm going to go off on some cheesy 70s movie reference. There was a movie called Logan's Run. And going back in the 70s, never trust anyone over 30 and all that, stuff like that too. Well, when people turned 30, they had like this gem that was inset in their hands and it would start to glow. That was the signal that, oh, you're old now. You have to go to like the suicide machine and have your body recycled or whatever, something like that too. So essentially our ubiquitin is, a sensor that says, okay, time for you to die. It's not a great movie, but it's an interesting premise. So ubiquitinated proteins get cleaved by this proteasome complex, which has 26 subunits. It's massive, and it chops them up. Ubiquitin is pretty much omnipresent in eukaryotes. Pretty much all eukaryotes have some form of ubiquitin. All right, and that's it for this chapter. Woof, right? There we go. All right, so that's not too bad, I hope. I don't think that's awful. Let's see if my don't know what I just closed, but it was something. There we go. Okay, so let's check out my. Oh, come on, man. Yeah. Okay. Because we only have one more class technically, where I'm going to give you the last exam, I want to go ahead and take a quick peek at the very last chapter. I know you guys are like so happy, like 5 chapters and we're burning through them. Okay. But I think what I'll try and do is, I've got the review sheet up for the final exam, but I'll try and post a little thing about what you can expect for the final exam as well too. The reason why I wanted to open up this last one and kind of go through it is because the very last chapter ties in our gene expression regulation in here. And it can be a little bit long. Come here. No, that's not what I want. Where did you go? We are in there, right? Maybe I'm no longer recording. Yes. Zoom, where are you?
Did you leave Zoom?
Maybe I did. No? Well, oh well, if I did, I did. If it's still recording, yes. Let's do it this way. This is why I like live streaming from home. No, it's still going. Okay, awesome, great. Okay, just can't find it when you got the projector going up here. Mutter, mutter, mutter. Okay. All right. So let's have this recognized stuff. I also learned something very important. Do not try to annotate PowerPoint slides while you're sharing a full screen in Zoom. It completely shut down, locked up my laptop. All right. So if we look at this last section in here, it looks fairly innocuous, but we do have, a long section here. It's because we're going to try and talk about a whole bunch of different examples, which I may kind of gloss over a little bit. But the idea here is we're going to take these last 4 chapters and kind of combine everything, just like we saw with our hormonal regulation in chapter 23, chapter 23. Our DNA elements control transcription, our protein factors control transcription. We have this example of the lac operon in there. I may not go into much detail about that. And we can look at our protein synthesis control by RNA. Okay, come on. Yes, there we go. So how do we regulate the protein concentration in the cell? Okay, there are lots of possible ways we can target this. It's kind of like, how do you build a house, right? Okay, well, it depends on what you want to do with it, where you want it to be. Is it going to be in some place very cold where you need good insulation? It's going to be some place at the beach where you want breezes flowing through, all that. There's lots of different ways you could do this. So we could target the synthesis of the primary RNA transcript. We could target processing it. We could look at post-transcriptional modifications of the mRNA, degradation of the mRNA, the actual protein synthesis itself, its post-translational modification, targeting and transport of the protein, and degradation of the protein. Right? So we've got lots of different ways that we can slow down or speed up protein metabolism to regulate its concentration in there. Okay, so 7 processes: transcription initiation, post-transcriptional processing, RNA stability, translational regulation, our protein modification, our protein transport, and our protein degradation. So we want to look at just the overall trends of this. A lot of what we focused on in the past has been transcription initiation, but it looks like post-transcriptional and translational regulation are something that the cells tend to do a lot as well too. So we can often have some really elaborate, I mean, if you thought regulation of the metabolism for like the TCA cycle or the urea cycle were crazy, you can just get lost in all this. All right. So let's just for today end up with a little bit of vocabulary and a little bit of definitions. Okay. So you often hear somebody talk about a housekeeping gene. And this is basically something that's under constitutive expression. And this is probably a sexist term that dates back, decades. You think, well, what do you need in the house? You gotta have an iron, you gotta have a broom, something like that. So all the stuff that you need for a house. It's like all the stuff that you need for the cell to just keep it active. All right. So a housekeeping gene is constantly expressed, usually at roughly the same levels in pretty much all cells. Okay. Many of these can be things like the histones for the DNA in the cells, some structural proteins and stuff like that. So housekeeping genes, there's not as much regulation because they're fairly constant. Okay. Regulated genes, you're going to see the levels of that gene rise and fall as the organism experiences different needs in here. So we can call these genes inducible because we can turn them on, and we can call them repressible because we can turn them off. Okay? So inducible, repressible, same thing. They can be regulated. It's just one is by turning them on and one is by turning them off. I like to think of the dimmer switch for the lights, which we don't have in this building anywhere, but you can turn up the brightness, you can turn down the brightness. So if we look at the binding of RNA polymerase to its promoters, that's a huge target of regulation. And there are so many promoters out there in so many different ways that these promoter sequences can be used that they encourage. I was gonna say it's like I'll use my good example of like the barker at the carnival. It's like, come see the bearded lady or something like that. They're out there trying to draw it in. Our promoters are going to want to get those RNA polymerases to bind. Okay. Um, and they're going to enhance the interaction in there, enhance the binding of the RNA to a sequence in here. You can also have a variety of transcription factors in there that can work to make it more likely that the RNA will bind or make it less likely. So you can inhibit or promote. I should use the vocabulary there because promoters are usually within the DNA itself, and transcription factors enhance or inhibit externally because they're not part of the DNA. Many bacterial promoters interact with the sigma factor of RNA polymerase. We didn't go into that much detail, but it's just like that's our signaling, sigma for signal, I guess is the way we use that too. But you got your Tata boxes in there. Those are the ones that are pretty much omnipresent, but you can have a variety of upstream elements as well too that will help interact with that sigma portion of the RNA polymerase to get it to bind. So if you use a bunch of different sigma factors, and a bunch of different things that interact with them, or if actually a bunch of different genes use the same sigma factors, then you can have coordinated expression. So rather than just turn on this one gene and have a different sequence turn on the next one, and different sequence turn on the next one, you can say, hey, we're going to turn on this entire family of genes. Okay, so we'll see that function in there. We can also see coordinated binding of these transcription factors to the promoters as well too. That can help tailor our specific response. So maybe we need more of one particular gene. We can allow expression of specific genes in response to environmental signals. Specificity factors. Yeah, that's what our essentially transcription factors in there. One example of this you can see in the heat shock. It'll induce transcription of new products to help protect the cell. If you look at bacteria, they like to grow at 30 degrees Celsius. You grow them at 37. You're going to see RNA polymerase replace the sigma 70 with sigma 32, which interacts with different promoters. You get heat shock proteins that are expressed, which help work to stabilize the E. coli cells at that higher temperature. Okay. And it always made me think, oh, they're cold shock proteins, because I could use some of those. Small molecules can regulate the activators and repressors. So repressors reduce interactions or block the polymerase completely. Effectors bind to the repressor and induce conformational change to either increase or decrease its affinity. So lots of different ways. We can either target the interaction, or we can change the structure of our polymerase or change the structure of our repressor as well too to alter its interaction at that binding site. Activators can improve the contacts in there. So it's kind of like guide them in there so that they fit better, and call these enhancers. Usually, enhancers will be adjacent to the promoters, and typically our promoters sometimes can be weak, so the activator helps make a little bit stronger interaction to promote that gene expression. For eukaryotes, the enhancers can be thousands and thousands of base pairs away because you've got all that folding in there in the chromatin structure and unfolding thousands of base pairs, but it can end up next to the polymerase to induce a reaction. All right. I think what I want to do for now is just kind of stop here because I wanted to find maybe a shorter version of this, but this is an excellent discussion. Anyway, but we'll pick up with that on Monday. We'll finish up on Monday, and I'll probably send out something later this week about the final exam, which will be similar to exam 3, which I still haven't finished because whatever, I'm just terrible. All right. Ugh. Let me see if I can find